στην αρχή.
Η χαρά είναι ελληνική, δεν
η χαρά είναι ελληνική.
Που βρίσκομαι εδώ να σας μιλήσω για κάποια από τα project για τα οποία δουλεύουμε στο εργαστήριο του Βασίλη. Okay, now I'm going to switch to English. So, our main interest is focused in the telencephalon, which is the most rostral part of the mammalian forebrain. And you can see here a coronal section of an adult mouse brain. And the mouse is the model that we're using for our studies. So by gross morphological criteria, we can identify two domains in this section, a basal one, which is the basal telencephalon, and a dorsal one, which is the cortex, which is correlated with cognitive function. And judging by the anatomy of this structure, it might seem that it's quite simple, but actually we know that there are quite a lot of different cell types that are found in this in the cortex. And as developmental biologists, the questions we want to ask is how all these different cell types are generated, how they interact with each other in order to make networks which is the functional base for the normal brain function. So in the cerebral cortex, there are two major neuronal types. There are the principal ones, which are the projection neurons. These neurons, they receive input from the periphery, and they are excitatory, and they use our neurotransmitter glutamate. And there is another neuronal population, the GABA-rich interneurons. These are inhibitory neurons. They comprise 20% of the total neuronal population of the cerebral cortex. And their function is to control the activity of the projection neurons and maintain a fine balance between inhibition and activation. And we know that this is very important because even when we have small alterations in excitation and inhibition, we know that in most of the cases this is due to dysfunction of cortical interneurons, and we have the development of seizures. And also there are quite a lot of neuropathies that are due to the dysfunction of cortical interneurons, and they are called interneuropathies, and among them is autism, schizophrenia, and even Alzheimer's disease. So I mentioned to you that cortical interneurons control the function of projection neurons, and they are doing this obviously by forming synapses. And they can form synapses in different subdomains of the pyramidal neurons, in the soma, in the dendrite, and this is one of the criteria that we are using in order to identify subpopulation of cortical interneurons. Because actually interneurons is a very diverse population, and we are using morphological criteria. We also use electrophysiological criteria as well as molecular criteria in order to subdivide them in different subpopulations. And for the rest of the talk, I would like to remember that there are three molecules, two neuropeptides, the VIP and the somatostatin, and the calcium binding protein, parvalbumin, that are expressed in three non-overlapping interneuronal populations. So there is another difference between the projection and neurons and the interneurons among function, and this is related to where these cells are born. So projection neurons are born in the cortex and they migrate radially in order to form all the layers of the mature cortex, while interneurons are generated in the basal brain in a domain that during embryogenesis is called ganglionic eminence. And they have to migrate in order to reach the cortex. First of all, they have to migrate tangentially, parallel to the ventricle of the developing brain, and then they have to switch their mode of migration to radially in order to populate the developing layers of the cortex. And actually, it has been shown by transplantation experiments that this domain, the ganglionic eminence, it can be further subdivided into subdomains, and each subdomain can give rise to different subpopulation of interneurons. And for example, in the most rostral part of the ventral telencephalon, in the MG, you have the generation of interneurons that they are going to differentiate it into the parvalbumin and somatostatin population, while in the caudal part, in the caudal ganglionic eminence, this domain is going to give rise to VIP interneurons. So this pattern is quite reminiscent of what is happening in the spinal cord, and therefore we and others thought that a combinatorial expression of transcription factors, which is going to be distinct in each subdomain, it might be important for the generation of the different subpopulation of interneurons. So I'm just going to focus on the medial ganglion terminals and describe to you the molecular cascade that we know so far. So in the medial ganglion terminals, cyclic progenitors express the transcription factor MTX2.1. When the cells exit the cell cycle, they express GABA, therefore they are differentiated and they are specified as GABAergic, and we also express the transcription factor LHX6 and SOC6. When they start to migrate in cortex, they don't regulate the expression of MTX2.1, but they maintain the expression of LHX6 and SOC6, and both transcription factors are still expressed in the adulthood in both subpopulation of NC-derived interneurons. So our interest was focused at the beginning in LHX6 because this is a transcription factor that has been identified in the lab. And in order to study the role of this factor in the development of cortical interneurons, we have generated a knockout mouse. And what we have seen by analyzing this mouse at postnatal stages, we know that the number of interneurons that are found in the mutant mouse is the same as in the control, but they are not properly distributed upon the layers of the cortex. But in addition, there is a defect in the differentiation of these interneurons into the parvallum and the somatostatin population. As a result, and as a further manifestation that there is a defect in the interneuronal network in the LHX6 knockout mice, what we have seen is that there is a decreased number of inhibitory currents in the LHX6 mutant mice, and we have shown this by performing a patch clamp in granular cells of the dental gyrus.
So I missed that point. So,
yeah.
Sorry.
Hello.
Okay, my question is, do they differentiate if they don't migrate? The cells?
If they don't
The post-mitotic cells, do they differentiate where they are, or they need to migrate in order to differentiate?
No, they need to migrate in order
They need to migrate.
differentiate, and they have to be in the environment of the postnatal brain. And I'm gonna focus actually into this in the rest of the talk.
Yeah, it's a total knockout. It's not.
This is a full knockout, yes, but I'm gonna briefly describe to you what's happening. But as I mentioned, the LHX6 is expressed only in interneurons. It's not expressed in projection neurons, so it's a cell autonomous defect what we see. Okay. Right. So, just to summarize what I've told you, LHX6 knockout mice have defect in the migration of these interneurons, in the distribution of interneurons in the cortex, and also in the differentiation of GABAergic interneurons into the two subpopulations that arise from the MG. So obviously the question that we have is whether this differentiation defect is due to the fact that these neurons, interneurons, they are not located in the proper position, or whether LHX6 has actually a role in the maturation and differentiation of these neurons. And in order to answer this question, we have generated a conditional LHX6 mouse. And these are experiments that we are currently doing. We are crossing this conditional mouse with different tree lines, and therefore we can specifically delete LHX6 at postnatal stages, and therefore answer this question of differentiation and maturation. But also we can delete LHX6 into the two specific subpopulations that arise from the MG, and therefore we think that we are going to manage to identify transcriptional cascades which are downstream of LHX6 and are important for the somatostatin or the parvalbumin differentiation. And another problem that we have with a full knockout mouse is that these mice, they are dying very young, and although I've shown you some experiments which suggest that they have a problem in the interneuronal network, Unfortunately, we cannot do any behavioral experiments, and therefore we cannot know even if they are developing spontaneous epileptic seizure. So the genetic models that we have generated, they survive until the adulthood, and therefore we think that they're going to be very useful to study distinct epileptic encephalopathies. And I'm just going to very briefly describe to you one of these conditional mice that we have generated. And this is a mouse in which we have deleted LH6 by using the FORTH's line, and we're deleting LH6 from the early stages. And although we would expect that these mice will also die very young, since an LH6 is deleted from the precursor state, these mice, they do survive. They do have problems in the differentiation of both subpopulation of MZ-derived interneurons, but on top of that, we see that we have cell death in the MZ-derived interneurons, and also we know that these mice, they develop seizures.
Is the defect, the anatomical defect, as severe as the full knockout, or is it lighter? The absence of parvalbumin is how to say.
It's exactly this. I mean, there are some differences. You have a massive decrease in the number of parvalbumin and somatostatin interneurons, but there is a difference that I'm going to show you in the next slide. So
this difference will also explain the difference in viability.
Not really. We actually don't know. I mean, the thing is that by using this NCH2.1 line, this is not a line which is expressed in all the MZ interneuronal precursors, so there are some escapers. But we are not sure if this is enough to rescue the lethality. But what I want to say is that we don't really think that the full knockout mice dies because of any neurological problem. We think that it has to do with additional problems which are related to the physiology of this mouse. So, okay, I just want to show you one of the first experiments that we are doing in order to observe the behavior of these mice. And this is a control mouse, which is very happy and goes around and explores the environment in the cage. And this is the mutant mouse. They have defects in the movement and in their motility, and also they have an extreme grooming. And these are signs that they are epileptogenic. So there is an additional reason that we are very excited to study the physiology, physiology and the behavior of these mice. And this is the fact that in these mutant mice, we have a number of parvalbumin interneurons that they are remaining in the mutant cortex, but these interneurons are completely malformed. They have an extremely large cell body and arborization, arborization. And we're excited because this type of parvalbumin interneurons, they have described that in a disease in human patients that they have developed epileptic seizures, and it's not really certain whether they are so big because they are trying to compensate for the defect in the reduced number of interneurons, and therefore they're trying to make as many synapses as possible with projection neurons, or actually whether they are part of the defective epileptogenic circuit of this brain. So, as I've mentioned to you, we are doing electrophysiological experiments at the moment. And finally, another aim that we have is to rescue the phenotype of these mice by performing interneuronal grafts. The way that we are doing this experiment is that we isolate interneuronal precursors from embryonic stages. We mark them by different methods, we dissociate them, and we directly inject them in the postnatal brain of mice. And I'm just going to show you a control experiment where we have sacrificed these mice after a month. And as you can see, these grafts, they are very happy and they survive in the environment of the host mouse. And on top of that, we know that they are differentiated in parvalbumin and somatostatin interneurons. So all these are experiments in progress, and I hope the next time I'm going to be here, I'm going to tell you about the results. So the fact that I'm using MZ interneuronal precursors and I graph them in the postnatal brain, and these interneurons, they manage to properly differentiate and integrate in the host network, might suggest that all that you need in order to make interneurons is just the battery of transcription factor which is expressed at these early developmental stages. But this is not true, and we know that this is not true because most of the maturation markers are expressed only in postnatal stages, and actually parvalum interneurons, they are not fully mature until the third postnatal week. So we hypothesize that there might be additional factors that are expressed later during development, and these factors that might be important for the maturation of cortical interneurons. And in order to identify these factors, we have performed microarray experiments in which we have compared the cortex of LHX6 and controls and LHX6 mutant mice. And one of the factors that we have identified to be downregulated in LHX6 mutant interneurons, that they are already in the cortex, is a transcription factor SADB1. So SADB1 is a typical transcription factor, and it can bind to the promoter of certain genes and control the expression. But in addition, it's a genome organizer. It can bind to specific DNA sequences. It can promote looping of the chromatin, and as a result, it can influence the regulation, can regulate the expression of multiple genes in the genome organizer way.
And so that's downregulated in LHX6 mice.
This is downregulated. So what it has been known is that SADB1 has been described in the immune system, and it plays a very important role for the maturation of T cells, but also has a very important role in breast tumor cells, where it has been shown that controls regulate the expression of more than 1,000 cells and promote the metastasis. So when we started working with SADB1, it hasn't been described whether it is expressed in neurons, in the CNS. So first of all, we wanted to figure out, obviously, whether it is expressed in GABAergic interneurons, and we used a mouse line where all the neurons are related with the GFP protein. And what we have seen is that SADB1 is expressed in cortical interneurons only when they are already in the cortex and they have stopped their tangential migration. And this expression has a very specific temporal sequence. So at early developmental stages, just a few cortical interneurons express SADB1, but actually then by the early and postnatal stages, all interneurons that arise from the HMG, they're going to express SADB1, and SADB1 therefore collateralized with somatostatin and parvalum interneurons, but not with the VIP interneurons which arise from the cotton ganglionoid, which is an LHX6 negative domain.
And vice versa, all SADB1 cells are interneurons?
No.
There are other kinds.
So just to summarize this part, we have identified a new transcription factor that is expressed in cortical interneurons that arise from DMG, but only when these interneurons are finished the tangential migration and they are located in the cortex. So we thought, because of this spatiotemporal expression of SADB1, this factor might be important to initiate a maturation program of cortical interneurons. And we thought that there are two major criteria that this factor should fulfill in order to consider it as a maturation promoting factor. First of all, it should stop the migration of interneurons, and then it should induce the expression of mature markers. So to investigate this hypothesis, we have performed electroporations in interneuronal precursors in the middle and lower terminals, and we cultured these cortical slices for 2 days in vitro. And when we electroporated the control slice, what we can see is that RFP interneurons, they migrate towards the cortex and they have the typical morphology of the tangential migratory interneuron. But when we overexpress SADB1 in these interneuronal precursors, these cells fail to migrate. On top of that, we wanted to figure out whether we have changed also the molecular character of these precursors that overexpress SADB1. So we performed the same type of electroporation, but this time we dissociated the electroporated domain. We have performed FACS sorting and then RT-PCR. And what we have seen is that we see a dramatic downregulation in markers that they express only in the middle ganglions, or they are involved in the migration of cortical interneurons, at least at that stage, while we have a significant upregulation in other markers that they are related with the maturation of neurons. We wanted to explore further whether these SADB1 overexpressing neurons can manage also to express finally differentiated markers. And indeed, we have seen that they have a dramatic upregulation at the mRNA of the somatostatin of NPY, although we don't see any major defect in the expression of KCC1, which is a gene which is related and is expressed in parvalbumin interneurons. So all these are differences at the mRNA level. What's happening at the protein level? Similarly to what I have described to you before, we electroporate the slice, and this time these dissociated cells, we place them on top of PZL feeder cells and we leave them to be differentiated. And after 2 days in vitro, what we can see is that the cells that we overexpress at B1, we have a massive upregulation in the number of somatostatin interneurons, and the same is for NPY. So these results suggest that SADB1 is sufficient for the maturation of the interneurons, and on top of that is sufficient for the differentiation of the somatostatin interneurons. So the question is whether...
But is SADB1 normally overexpressed in this process?
No, but that was the point of the experiment. The point was to overexpress it at the stage where the neurons they are immature and they migrate, and what we have seen is that we block the migration and we force them to differentiate and adopt markers that we find only in mature interneurons.
And this you consider an indication of its normal function, even if you have overexpression. In other words, you don't express it at normal level.
No. I
You
don't
express it
express
higher than normally is expressed when it's doing what it's doing.
Yeah, but what I have shown is in agreement with what we see actually in vivo. First of all, that we don't have SADB1 expression in migrating interneurons. You see upregulation of SADB1 expression only when these interneurons are in the cortex and they stop their tangential migration, and then they start to differentiate. At that stage, at T15.5, where SADB1 is expressed, we see also for the first time the expression of this marker KCC2, which is a channel which is very important for the maturation of interneurons. And yes.
And you're going to tell us what's happening.
What?
You're going to tell us what's happening over the
Of course. But first of all, I'm going to show you what's happening in vitro in these cultures, where we overexpress an siRNA construct for SADB1, and what we see is that we don't get any somatostatin interneurons. So these are in vivo experiments. So what's happening in the in vivo situation? We have generated a full knockout SADB1 mouse, and what we have seen is that we have the same number of GABAergic interneurons in the cortex of this mouse, but we have a massive decrease in the number of somatostatin interneurons, and parvalbumin interneurons, they are still affected, but to a lesser degree.
So what is the relationship with LH6?
I'm going to say...
Okay.
So, what I've shown you so far is that SADB1 is sufficient and necessary for the differentiation, the maturation of interneurons, and is very important for the differentiation of the somatostatin subtype. So exactly the question is what the relationship between LHX6 and SADB1. So first of all, we've checked our knockout mice, and what we have seen is that in LHX6 knockout mice, you don't have expression of SADB1. And in order to figure out whether SADB1 and LHX6 work in the same transcriptional cascade, what we have done, we overexpress SADB1 and want to figure out whether we can rescue some of the effects of the SADB1 LHX6 mutants. And this is exactly what we have seen. Now, when we are overexpressing SADB1 in LHX6 mutant cells, we manage to rescue a version of SADB expression. This experiment suggests that LHX6 and SADB1, they work in the same transcriptional cascade, but the question also that arises is LHX6 sufficient for the induction of SADB1? To answer this question, we have changed our experimental procedure. So actually what we are doing, we are performing in utero electroporation in cortical progenitors. So these cortical progenitors, they're giving projection neurons and not interneurons. But when we co-electroporate LHX6 and MASH1, we manage to generate interneurons. And actually we see that quite a lot of the markers that we have identified in our microarray experiments as potentially downstream targets of LHX6, they are upregulated, and I'm showing you here this example. Unfortunately, that was not the case for SADB1. So when we overexpress LHX6 in cortical progenitors, we don't manage also to induce the expression of SADB1. Therefore, SADB1 works in the same transcriptional cascade with LHX6, but LHX6 is not sufficient on its own to induce the expression of SADB1. So that would suggest that potentially another transcription factor, together with LHX6, can induce the SADB1 expression, but also it could be a completely different physiological mechanism. And we thought, because SADB1, as I mentioned to you, is expressed during late developmental stages, and we know that during these late developmental stages, we have cells that are active and have spontaneous activity. Potentially, this spontaneous activity might be important for the induction of SADB1. So, and SADB1, it might be an activity-dependent gene. So when I'm talking about activity-dependent genes, very simple, you have a signal that causes the depolarization of the neuron, and as a result, you have the activation of voltage-gated calcium channels, you have influx of calcium into the neuron, and these trigger certain signaling cascades, which are going to lead to the activation of new genes, which are called activity-dependent genes. Now, in order to mimic this phenomenon, we have developed cultures we culture our cells in the presence of potassium chloride. As a result, we have the influx of potassium, which again causes depolarization of the neuron, and then the same physiological cascade is activated. And the question is whether we are going to manage to see upregulation of SADB1. So first of all, we wanted to figure out whether our cultures can mimic the temporal expression of SADB1 that we see in vivo. So we dissociate the cortex of V14.5 mice, and when we plate the cells after 1 day in vitro, we see that we have very few number of neurons that express SADB1. After 2 days in vitro, this number is increased, and after 3 days in vitro, the number of interneurons that express SADB1 is 70%. So now we culture these cells for 2 days in vitro in the presence of potassium chloride, and what we can see is that we have a massive upregulation of FORTH's, which is a known activity-dependent marker, but at the same time we have a very significant increase in the expression of SADB1. And to further investigate that this is due to the physiological pathway that I have described to you, and that these calcium channels are implicated, we performed some basic pharmacological experiments in which we block the activity of this calcium channel, so therefore we don't have influx of calcium into the neuron. And what we see when we culture our cells in the presence of KCl, but together with this inhibitor, FORTH's, we see that we are reducing the number of interneurons that express SADB1 at the control level. And we wanted to figure out which is the signal that actually can trigger in vivo this phenomenon. And we wanted to figure out whether ambient GABA, which is actually the GABA which is released from the cells, it might be important for the induction of SADB1. So we have blocked GABA A receptors, and we have seen a significant, although small, reduction in the number of interneurons that they express SADB1. So overall, what we are suggesting is that LH66, together with spontaneous activity, is very important for the induction of SADB1 in cells which are already located in the cortex, and the expression of SADB1 is important to induce a maturation program for both subtypes of MHC interneurons, but in addition is very important for the differentiation of the somatostatin interneurons.
But do you actually know that, let's say, this is not happening in LH6 knockout cells, the induction of SADB1, or you can block it with siRNA against the stimulation, the stimulation triggered transcription of SADB1? Does it require? Yes. Because? How do we know that?
Because I have done the experiment in LH6 mutant cells, and I couldn't see an upregulation.
But do the cells maturate to any level that they could respond to the stimulation of the?
No.
Right.
Actually, they don't really upregulate these marker cases.
Right.
Yeah.
So you don't really know if it's required for the expression of SADB1 or for the cells to maturate in a state that express SADB1.
It's
But
required directly for the expression of SADB1 or indirectly.
Right.
Which one? The LH66?
The LH66, yes.
No, LH66 we don't have any results, but they suggest that directly binds to the promoter of SADB1.
No, it could be indirectly, but.
Or indirectly.
But does it, well, I guess what I'm asking is, do you need to express it at the time to have it, prescribed and present as protein at the time that SADB1 starts expressing? Do you need it at this very moment, independent if it's happening directly or indirectly? In other words, if you block it with siRNA in your culture, what would happen? Are they going to respond to the cells?
Okay, I haven't done this experiment, so I haven't done an acute deletion of the expression of LH66 and see what's happening. I have used LH66 mutant cells, which obviously they are mutant since the time they are born. So yes, I mean, this point might be not clear, but I mean, the point that I want to make is that usually we have in mind that we have transcription cascade and one transcription factor together with someone else and use something else. All I'm saying is that in this situation, you have a different pathway, and this is a physiological pathway, and this pathway you can find it only in this specific area of the brain because there are other specific characteristics. So, for example, in order to respond to the ambient GABA or glutamate, which anyhow exist, these immature sections and neurons, I mean, you need also to have increased number of GABA receptors or different units of GABA receptors, and we don't know how this is controlled. So all I'm saying is that maybe LH6 is required for, I don't know, the different GABA receptors that are expressed at this specific state, and this is important for the whole physiological pathway to activate SADB1. But that's how maybe LH66 works.
So it looks like it's indirect, but what you're saying is that perhaps LH6 drives some sort of differentiation or maturation to a
certain point.
To a certain point
that we
where
don't really know.
spontaneous activity is sufficient to drive SAB1 expression, something like that.
Together, these two.
Yeah.
Because in the AIDS it's excluded
Together.
time, so you need both. Okay. Right. So, okay. So, but with what I have described to you so far, I've told you that there is a dramatic loss of somatostatin interneurons, and what you would have expected is that you have a defective inhibitory circuit in these mutant mice, and these mice they develop generalized seizures, and we would expect something like that because in the lab we have generated another genetic tool, another knockout mouse. In this, we downregulate the expression of LH6 is downregulated by 30%, and we have a 30% reduction in the number of somatostatin interneurons. And these mice, they develop spontaneous epileptic seizures. But this is not what we see in our SAB1 knockout mouse. And so far, I was emphasizing the expression of SAB1 in cortical interneurons, but actually SAB1 is expressed in projection neurons. And the knockout mouse that I have described to you so far has deletion of SAB1 both in the projection and in the interneuronal population. So we thought that we might see a more obvious defective interneuronal inhibitory circuit if we specifically delete SAB1 from cortical interneurons. And therefore we have generated knockout mice by using this NKX2.1 preline that I have described to you previously, and by which we delete the SAB1 from all NCE-derived interneurons. So these are experiments that have been performed from Melanie Kalaitsidou, which is a PhD student that she was working with me, and she's still in the lab, and she's gonna finish these experiments that I'm gonna describe to you now. So what we see in this conditional mouse, in this mouse where SAB1 is deleted only from the somatostatin, only from the interneurons, we see again a dramatic loss in the number of somatostatin interneurons and a significant loss in the number of parvalbumin interneurons. But most interestingly, these mice have loss of balance, muscle twitching, and they have this phenotype even after mild handling. And on top of that, we see a significant cell death in the number of NCE-derived interneurons, which is in correlation with the fact that these mice they might be epileptogenic. But most importantly, what is happening is that we see a massive upregulation of FORTH's in projection neurons, and also another regulation of SAB1 in projection neurons as well. So what that means is the fact that the projection neurons are hyperactive, and therefore this might be the cause for the epileptic seizures. And actually we know that this relationship between SAB1 and FORTH's, the fact that we see upregulation of SAB1 is correlated with an upregulation of FORTH's, this is actually a direct event, because another lab has recently shown that SAB1 binds to the promoter of a lot of activity-dependent genes. And among them is FORTH's that we see that this upregulator in our mutant mice. So therefore we have a transcription factor, SAB1, which is expressed in both populations, and by manipulating the expression levels in one or the other population or in both populations, we can generate mice that have a completely different network and therefore different behavior. So
did you say, and I missed it before, that SAB1 minus inhibit migration as well?
It's not expressed in my
so it's
expressed only when they
so it doesn't inhibit migration, it inhibits only the functional.
Yes.
And when you say levels, you mean all or nothing, or the heterozygous phenotype?
I don't know. It doesn't happen. Which
When you say manipulating the levels, you mean all or nothing?
in that case,
Yes.
no, I mean all or nothing in
Okay,
that case.
so it's not.
Yeah. Delete the expression of SAB1 in one of the two populations. So when we delete SAB1 in interneurons, what we see is that we have a defect in the maturation and the survival of interneurons, And on top of that, we have an upregulation of SADP1 and C4s in projection neurons, and that causes an imbalance between excitation and inhibition, and therefore these mice develop seizures. So now what's actually happening in the full knockout mouse, we have deletion of SADP1, both in the interneurons and the projection neurons, and interneurons, they have maturation defects, and both cortical neuronal populations, they have the downregulation of certain activity-dependent genes. So therefore, we have both populations that they are not normally function, and therefore we don't have the development of seizure, and we don't know if there are any additional defects because these mice again die quite young, and therefore we cannot perform proper behavioral analysis. But we are very interested to figure out what actually is going to happen when we are specifically delete SADP1 from projection neurons, and therefore we have generated mice in which we have deleted SADP1 from progenitor neurons by using the next tree line, which is expressed in post-mitotic pyramidal cells.
Sorry, the one on top is combined knockout, right?
Conditional.
Conditional, but it's both SADP1 and NKX2.
No, the NKX is the tree line that we are using to delete SADP1.
Okay.
Yeah. Okay. So I just want to finish with a working model that we have about the role of SADP1 and its importance in brain function. So we think that SADP1 is an activity-dependent gene. So upon neuronal activity, you have calcium waves that you have the influx of calcium in the neuron, and this trigger signaling cascades that are important for the activation of new genes. And these genes are activity-dependent genes, and among them is SADP1. So the expression then SADP1, then SADP1 combines to the promoter of these activity-dependent genes with a typical manner with the function of a typical transcription factor, but also can modulate and regulate the expression level of these genes through this genome organizer function that it has. And we know that when all of these activity-dependent genes, they are implicated in a lot of functions which are important for the development of the central nervous system, such as neuronal survival, dendritic growth, and synapse formation, and therefore is very important for the maturation both of the interneurons and the projection neurons. And we also think that a similar pathway and a similar role has SADP1 in the adult brain. And we have the same physiological cascade, but in this time, SADP1 might be very important for synaptic plasticity, and therefore for the generation of memories and learning. And actually, there is quite a lot of interest in order to investigate whether humans have mutation in this transcription factor, which are correlated with certain neurological disorders. So with this, I would like to finish by thanking Vasilis, which gives me the opportunity to work in his lab, in a project which actually is not the focus of his lab, current members of the labs, past members of the labs with whom we are still collaborating. So all the electrophysiological experiments I'm doing them with the help of Guillermo, who is now working in the lab of Juan Burone, which is in the MRC unit of King Storage. There is also a project that I haven't talked to you about that is related with the role of interneurons in the patterning of the direct cortex. And I'm doing this project in collaboration with the neurophysiology department and Trauma Green, obviously Rona, with whom we are collaborating in the migration of interneurons. So the first microarray that I have described to you, and this is the microarray by which we have identified SADP1. It has been done with Horizon Genomics, and all the RNA-seq experiments that I'm currently doing are with the help of the high throughput sequencing service in the NMR. And also I would like to thank collaborators that have given me certain tree lines These lines have generated from the lab of Josh Huang in Spring Harbor, but now they are available from Jackson Laboratory, and also the biological services which they take care of these several lines that are maintaining in the animal house. So thank you for your attention.
Questions?
Yes.
A simple one. Did you test the effects of somatostatin in the expression of all three transcription factors driving this phenomenon? The idea is if the early appearing somatostatin interneurons can amplify this phenomenon by just going back and increasing the expression of these transcription factors.
Can you just explain to me what you're suggesting?
My suggestion is that early appearing somatostatin interneurons can, by the phenomenon, amplify the phenomenon of differentiation of precursors by just regulating the expression of the transcription factors driving the phenomenon of differentiation and migration of this.
So your idea is to put in the area where you have interneuronal precursors more mature neurons and see whether this is going to influence the expression. No, haven't done an experiment. I have done exactly the opposite. I have done, I'm overexpressing actually SAP1 in these interneuronal precursors, and I'm just injecting in the other brain and see what is happening. And what I see is that because I'm blocking the migration, although I managed to make somatostatin interneurons, these are not functional because they have not properly integrated into the layers. But the opposite experiments now have done to see whether I might influence the maturation of this. It's not certain that it can be done. Yes, because as I told you, you need a certain environment, and just by adding a few cells might not be enough to change the environment where this interneuronal boost them to us. But
Here, a couple of questions.
yeah.
One is related to SAT2. SAT2, as far as I remember, it has a different expression pattern. It's more or less.
SAT2 is expressed only in progesterone.
In congenital
Yes.
neurons. So,
and also SAT2 mice, they die at P0, and it has been shown that it has this mouse that have this mice that have the defect in the specification of progesterone neurons and not maturation.
so there's no redundancy at all. There's no oral compensation of one for the other. No. The other is related to, perhaps what is the role of SAT1 neurons. Do I get it right that we have an activatory gene that expressed in inhibitory cells? Is that correct? We have inhibitory cells in the neuron, which expressed in activatory gene. SAT1 activates FORTH's.
It's an activity-dependent gene.
Activity-dependent gene that positively controls activation, right? No.
Excitation.
No.
Excitation.
And
It doesn't control that excitation because
No,
it has a lot of downstream genes which are what?
these are activity-dependent genes, which are related, for example, with dendrite formation and with survival, and these are the same both for progesterone neurons and interneurons.
Okay.
So it's a different. It's not that, for example, now these neurons they're going to express glutamate to make synapses, and they're going to activate other neurons. They always express GABA, and they make inhibitory synapses. Apart from early stages where GABA actually is excitatory, that's why you have. Yeah.
Okay. So why is it so specific in these particular cells?
They are in both. It is expressed in both cells, both in interneurons and projection
Okay.
neurons.
What about the cortex?
Only in the cortex. Why? Actually, it is expressed not in the hippocampus, only in the cortex, in the beautiful cortex, and it's not expressed at all, for example, in this triad. Why? I don't really know. Yes. But it is quite surprising the fact that, as I told you, the progenitors for cortical neurons and the progenitors for interneurons, they are actually from two completely different domains. Nevertheless, the cells that they end up in the same area, both of them express SAB1, and SAB1 has the same role in both of them, which is the maturation of both neuronal types. Yes.
What is the link to synaptoplasticity? You mentioned the
Yes. So what we have seen and others have described is that they have abnormal deadlines, the projection neurons, but this is happening only in the cortex and not in the hippocampus, because SAB1 is not expressed in the hippocampus apart from the subpopulation of somatostatin neurons. So again, this was the full knockout mouse, and the analysis now we are doing this condition on the extreme mouse is obviously we're taking the dendrites and also synapses. And all I want to say is that the next three mice, the mice that we did SAB1 from the projection neurons, they don't have epileptic seizures, and they have different phenotypes. We haven't fully characterized all of them. Then we know that they are aggressive, and we think that this is related to the expression of SAB1 in the amygdala. But
You showed very nicely the involvement of calcium channels. Do you have any idea how the calcium, the intracellular calcium does what it does?
I mean, I'm very
Is
sure.
there,
Yeah, it's a signaling molecule, and also when you have increased in the weights, the increase of calcium, you have also activated CMP, and we know that these transcription factors that are upregulated upon activation, they have certain domains which are serum response domains or calcium binding response domains. So actually, CREB is one domain, transcription factors that binds to the promoter of these things, and this indicates the induction of the expression factor.
there's nothing else in between.
It's a whole signal. Yeah.
And they respond to calcium blockers, I guess.
Sorry?
And the system responds to calcium blockers.
Great.
I've done this right? Okay. Thank you.
That's it.
Thank you.